My name is Lawrence Rosenberg, and this is the Alpha Human Podcast. Welcome back, everybody. And before we begin, let me kick things off as usual by stating that I am definitely not a doctor, nor am I a medical or healthcare professional. And the content of this podcast and any of the information, advice, opinions, or statements within it are definitely not intended and are not to be used as a substitute for professional medical advice or for diagnosis or treatment. And you should not disregard or delay in obtaining medical advice for any medical condition you may have, and you should seek the assistance of a healthcare professional for any such conditions. And always inform your doctor of any changes that you make or are thinking about making to your lifestyle. All right, so my people, in the last episode, I mentioned that since I've been dealing with chromophobe renal cell carcinoma, again, that's a form of kidney cancer. Since I've been dealing with it over the past 7 years, there have been two specific instances where the cancer had spread, once to another spot on my right kidney. I had the left one, by the way, removed. For anyone who hasn't been following me or heard previous episodes, I had the left kidney removed along with a very large 14-centimeter tumor just about 7 years ago. For all those who are tuning in for the first time, I had what was essentially a grapefruit-sized tumor removed from my left kidney, along with the kidney itself. And back then, by the way, they also had noticed at the same time a small 2-centimeter lesion in the center of my right kidney. And given I have only one left, my strategy, even though they noticed that there was this lesion, would essentially look like the same exact thing, sitting in the center of my right kidney, my strategy was that, hey, I'm not going to mess with it. I only have one kidney. Let's just watch it. Let's monitor that lesion. Take the left kidney. Let's leave the right one alone for now. And my plan of attack was, at that time, to only do some type of intervention. And for my research, that intervention would ultimately, if I need to, be what is known as an ablation, where they stick a needle in it and either burn it out or freeze it out. So a cryoablation or a heat ablation, an RF ablation, only if it were absolutely necessary. And to do whatever I could do on my end, nutrition-wise, ultimately, that's most of the control you're going to have is nutrition-wise. There's a few other things you can do, right? But I'll do whatever I can do on my end to stay healthy and heal myself. And I'll watch, what's going on in the right kidney, in the center of the right kidney, just make sure everything stays stable. And if not, if this thing starts growing, again, 7 years ago now, they found it, right? We'll just watch that thing. And so I've done whatever I could to stay as healthy as possible. And maybe, again, I'm boxing with cancer, right? So you're ducking out of the way, you're throwing a jab here and there, not necessarily looking for a knockout punch. Would be great if you could just knock the shit out of cancer and kill it once and for all. But again, I'm dealing with one kidney, right? So I'm trying to be really, really careful with this. So a lot of what I've done as I've monitored this lesion, this tumor in my right kidney, a lot of it has had to do with nutrition. Over the past 7 years, I've been on a ketogenic style diet, which over time evolved into a carnivore diet, which over time has kind of morphed into carnivore with just recently a little bit of fruit, a little bit of honey here and there. I've also been in and out of fasting protocols, which, based on my deep research, it turns out that, listen, as much as fasting, especially extended fasting, can be helpful in reducing tumor size and also as a protective strategy, by the way, if you were to be taking chemotherapeutic drugs or radiation therapy, there have been studies, if you go on extended fast, typically 3-day fast, before you go into chemotherapy or radiation, you can help keep your non-cancerous cells as healthy as possible and as strong as possible, while really helping to weaken the cancer-infected cells And this way you keep the healthy cells strong, right? Because you've fasted for 3 days, they're far more resilient. And then when you bombard yourself with some kind of toxic poison, you're protecting those cells because they're stronger than they would be had you not fasted. And there are studies on this. And so, when you're on these extended fasting protocols, there are some benefits. But the thing that most proponents of fasting are not aware of and never talk about is that depending on the type of cancer that you have, prolonged fasts can do the opposite of what you're intending and actually promote tumor growth. As some cancers, turns out like the one I have, and I've done some pretty heavy duty fasting protocols. I've done fasting protocols where for months, I would do 3 days straight of fasting every week, or I'd do 2 48-hour fasts per week, right? And I've done 5-day fasts, and I've done 6-day fasts, right? But it turns out, which is why it's so important to do your research, people, some cancers like mine actually use the autophagic process, which is what happens when you go into extended fast. You go into autophagy. And when you're in autophagy, this process is where you start recycling a lot of the dead and senescent cells that you have, the broken cells. But some cancers like mine actually use the autophagic process to absorb those dead, broken, and senescent cells that then get converted into their base substrates, which are those broken cells that are going through the cell recycling process. They get broken down into amino acids, into lipids, and into sugars. And those cancer cells, again, like the type of cancer I have, actually uses those cells to essentially cannibalize them for nutrition. Interestingly, even cancers that because most cancers primarily rely on accelerated glucose and glutamine metabolism for their nutrition. Well, it turns out that even those type of cancer cells, which are by and large the majority of cancers, actually rely on glucose and glutamine metabolism, ramped up, accelerated glucose and glutamine metabolism for their nutrients. However, those cancer cells can also adapt, and resort to cell scavenging and recycling mechanisms like phagocytosis, macropinocytosis, and endocytosis. They can start to rely on those processes when they're faced with nutrient scarcity or nutrient deprivation due to fasting protocols or due to drugs that inhibit glucose and glutamine absorption. And so this alternative approach that the cancer cells can take allows them to acquire the necessary resources to survive and multiply. This is why, people, it's very important to do your research, because you'll see all these videos out there and hear all of these experts talk about fasting and how it's a great strategy for cancer and for reducing tumor size. And listen, in many cases, it actually is. But you got to do your homework. You got to understand as much as possible about the type of cancer you are dealing with, because if you have a cancer like mine, and there are a few other cancers out there that primarily they don't rely on sucking in as much glucose and glutamine as they can. And so you go on a fasting protocol trying to limit that shit, trying to limit glucose, trying to limit glutamine. It doesn't really matter because those cancer cells are actually, they're not getting their nutrition with that type of ramped up metabolism for glucose and glutamine anyway. What they're doing is they're using the cell recycling processes that they have, the cell scavenging processes that they can actually rely on for their glucose, for their glutamine, right, for their lipids. All right. Anyway, another strategy that I've employed, by the way, while we're talking about strategies, again, I've got this tumor in the center of my right kidney, right? So what am I going to do about it? If I'm not going to take it out, number one, I'm going to use nutrition to keep it at bay, right? To keep it from growing. Another strategy that I've employed is visualization and using the mind to heal the body. And I'm going to absolutely do an episode on this, as it is a very powerful way to take control of your health in addition to your nutrition. This is a major way to go beyond just being a spectator or hapless bystander in the process of healing, where you simply just take the drugs your doctor prescribes and everything else is kind of left to the wind, and you're kind of blown around like a leaf on a breezy day, hoping that at some point you end up on safe ground, right? You just get a bunch of instructions from the doctors. You follow the protocols that the doctors give you. And you're just like this, innocent bystander, kind of like, okay, everything else is out of my hands. And, you should listen to your doctors, but you should also research what the doctors are telling you to do and get second and third opinions. Make sure you're completely dialed into every aspect of your healing process. But what I'm saying is, you got to take some control as well. You got to be invested in the process and do whatever you can do to heal yourself. So by using visualization techniques, you can exert control over the process as well and make a profound impact on your mindset, on your outlook. Because a lot of people, you have cancer, you have a very grim outlook. I know, because as soon as I found out I had cancer 7+ years ago, man, I went down a spiral. It's definitely not a good mindset. I snapped myself out of it within a day, right? I went back to my usual positive mindset, but for an entire day, I went down some ugly rabbit holes. But using visualization techniques, you can absolutely influence your outlook and your condition, and you can influence the healing process. And there are some great books on how to do this and some powerful strategies that you can employ. And we're going to talk more about that in one of the upcoming episodes. We'll see which one I do. It might be the next episode or maybe the episode after that, but I'm definitely going to dive into this. And then there's the biggest strategy of all, which is recognizing Jesus Christ as your Lord and Savior, handing things over to God and getting deep into prayer and gratitude for how blessed you actually are, how blessed I actually am, and starting your day off with prayer and communicating with the Lord, as I like to say. Jesus Christ empowers me with the strength, with the discernment, with the presence of mind and the guidance I need to heal and to live with passion and purpose. And so, look, with all of that said, I went off on a tangent, as I often do. Let me take a sip of water. If we go back to how I began this episode and also how I began the last episode, if we go back to the 2 times that my cancer actually spread, again, I've got this tumor sitting in the center of my right kidney, and I've been doing everything I can to just keep it there and also keep it from growing as much as I can do. But there's been 2 times in the last 7 years where that cancer has spread. Once to another location on my right kidney, as I've mentioned. That was about 4 years ago. And much like the lesion that I still have in the center of my right kidney, that one that now sits on the outside, the upper pole of my right kidney, is also now roughly 2 centimeters. It kind of grew to about 2 centimeters. It happened real quick too. On one of my 6 monthly scans, it just popped up at 1.1 centimeters. And then over a period of another year or so, it grew to about 2 centimeters, a little over that. And, I've kept it in check since then. It's kind of stabilized around that point. So I've got one in the center of the kidney and one on the upper pole on the outside of the right kidney, both roughly around 2 centimeters. And we continue to monitor it. No interventions, as I've said, unless absolutely necessary. And I'm doing what I can, as I just described, to keep it in check. But then in the second instance that the cancer moved, it went to my liver, right? And that one, we had been following it. We noticed it was noticed, at some point when it was about 7 millimeters, then it got to about 1.2 centimeters, and then ultimately got about 1.8 or 1.9 when they took it out of me, because that one I was having taken out, that's for damn sure. We had that taken out when it was about 2 centimeters, and we had that surgery last August. And because I am on such a disciplined routine and such disciplined nutrition program, I was able to look back and isolate any specific changes that I had made to my diet, to my program, that perhaps correlated with these two instances where the cancer had spread. And sure enough, I was able to identify some significant additions that I had made to my diet that preceded both of these instances. So the first time the cancer spread, when it moved to that other location on my right kidney, for about a year in the run-up to finding that lesion, I had added dairy to my diet in the form of cheese and a bit of heavy cream. Now, the heavy cream was only in my coffee, and so there wasn't a prodigious amount. But I did make cheese a staple of my nutrition program, and it certainly, look, it kept me on a ketogenic protocol. But I thought to myself, did that have anything to do with the emergence of this new lesion at the time? So I dove into the research, and what I found out blew my mind. And once I learned what I had uncovered, I stopped eating any form of dairy. And that's what we're going to discuss today. We're going to look into that today, right? The other major addition I made to my diet, and this was later on, prior to the second instance where the cancer had moved, this time to my liver. The only major discernible difference that was that I had added peanut butter to my nutrition regime. Now, I mentioned in the last podcast that I had also added for a period of time, not a long time, but there were, I don't know, for probably about 3 to 6 months, can't remember exactly. I also added some collagen protein to my diet, which is really, really high in certain amino acids. It's not a full spectrum protein with a full spectrum of, all the essential amino acids. In fact, collagen protein was very high in a amino acid called glycine. And so, it's unlikely, not impossible, but it's unlikely that the glycine increase is what caused the tumor to metastasize. It could potentially be a factor in maybe tumor size increasing, perhaps. However, when it comes to a tumor metastasis, right, the big thing here that I uncovered was peanut butter, right? And I had added a lot of peanut butter to my nutrition regime. I made it a staple of my diet, which again was still keeping with my ketogenic approach. Peanut butter is low in carbs, high in fat, with a decent amount of protein. But who would have ever thought that peanuts could have anything to do with cancer? Now, with cheese and milk products, there has always been some sort of conjecture and rumors and information that has from time to time broken through the mainstream press and the collective consciousness, such that if you pay any attention to food and health, you might have heard something about dairy being an issue with respect to cancer. But peanuts or peanut butter? No way. I never heard anything even close to suggesting that peanuts might be linked somehow or some way to cancer. Nonetheless, because that was really the only other significant change I had made to my diet, and it wasn't for 3 months, and it wasn't for, it was for quite a while, maybe a year and a half, about a year and a half worth of time that I had added peanut butter to the diet. So I really dove into the research on that, and I was stunned by what I found. And that is that while peanuts do not appear to cause cancer, God forbid, should you happen to have cancer, then based on my research and the evidence presented in a number of studies, which we talked about in the last episode, you better proceed with caution if you have cancer. If you have cancer and you have added peanuts to your diet, proceed with caution, because there's a protein in peanuts that has been shown to cause cancer to metastasize. That protein is called peanut agglutinin. It's a lectin. It's a plant lectin. It's found in peanuts. That was the subject of the previous episode of the Alpha Human podcast. And by the way, as you can imagine, I no longer consume peanuts or peanut butter. But if you haven't listened to that episode, listen to it. If you know someone who has cancer, send that episode to them. Warn them. Beware. If you got cancer, in my opinion, right? I stay the hell away from peanut butter. You might want to consider doing the same. However, in this episode, we're going to dive deep into dairy. and why, much like peanuts, if you have cancer, then you need to be warned. Right? You need to be warned because bovine milk, that's cow's milk for the uninitiated, bovine milk has been shown to cause cancer to metastasize. It's been shown to cause cancer to spread. So let's dig into why that is. Now, first of all, it's important to note that bovine milk is one of the most commonly consumed types of milk worldwide, and it's a rich source of essential nutrients and bioactive compounds. Right? So listen, if you don't have cancer, then fine, have at it. Milk and milk products such as cheese, yogurt, made up of a ton of a complex mixture of nutrients, and including proteins and fats and carbohydrates and vitamins and minerals. But with that said, and we could dive into the aspects of the different the carbs that are in there, the types of fats that are in there. But let's just focus for now on the different proteins that constitute a significant portion of the composition of bovine milk. And it's these proteins. The reason I want to focus on them, it's these proteins which have been shown to have both positive and negative effects when it comes to cancer. So let's delve into the protein components of cow's milk and see what we've got. Let me just have a sip of water here. Now, the major protein, the big one, when it comes to milk, is casein. Caseins are the major group of proteins in bovine milk, accounting for about 80% of the total protein content. They exist in the form of large aggregates called micelles. The three main types of caseins found in milk are A casein, B casein, and K casein. These proteins are rich in phosphorus and calcium, and they are responsible for the white color of milk. And the other major player is whey protein, which most people have heard of. You've no doubt heard of whey protein, especially since it is the main ingredient of almost all the protein drinks that are on the market today. Whey proteins make up the remaining 20% of the protein content of milk, and they include several different types of proteins. So whey proteins are made up of B lactoglobulin, A lactalbumin, lactoferrin, and immunoglobulins. In addition to these proteins, whey also contains other proteins in smaller amounts, such as serum albumin, glycomacropeptides, and other minor peptides. They are all soluble in the whey fraction of milk, in the whey percentage of milk. So all those proteins I've just mentioned, they're soluble, which means that these proteins have the property of being able to dissolve or disperse in the liquid portion of milk. Whey is the watery part of milk that remains after milk coagulates and curdles during cheese or yogurt production. If that's the case, then as you would imagine, the curdled and the coagulated portion is casein, right? So whey proteins, by the way, are also known for their very high biological value and are rich in essential amino acids, which again is why they're often the protein of choice for protein supplements, for protein bars, for protein shakes, etc. And when you drill down into them, again, as I mentioned, you've got proteins like B-lactoglobulin, which is one of the most abundant whey proteins in cow's milk. It belongs to the lipocalin family of proteins, and it's a source of essential amino acids. B-lactoglobulin has been studied for its potential allergenic properties in some individuals, so that's where you got to be careful of it. But then you have A-lactalbumin. A-lactalbumin is another whey protein that's rich in essential amino acids, particularly tryptophan. And it plays a role in lactose synthesis and is also considered to have potential health benefits. Lactoferrin, right? That's a multifunctional protein with antimicrobial properties. It helps bind and transport iron, which contributes to its immune modulating effects. Then you have immunoglobulins, which are also known as antibodies that are present in small amounts in cow's milk. They provide passive immunity to newborn calves by transferring maternal antibodies from the mother's milk. Bovine milk also contains several enzymes, including lactoperoxidase, lipase, and alkaline phosphatase. And these enzymes play roles in various biochemical processes, such as the breakdown of fats and protection against bacterial growth. Now, in addition to the major protein components, bovine milk contains mixtures of minor proteins together with peptides that contribute to its nutritional and functional properties. These include proteose peptone fractions, which are mixtures of proteins and peptides that are produced during the enzymatic digestion of casein. Again, as we know, that's like the major protein in milk. And those, those proteins and peptides, those smaller mixtures, have been shown to be associated with immune modulating effects. And milk protein can also be enzymatically broken down into smaller peptides, right? So you got the proteins, and then which are the amino acid chains, and you got the smaller peptides, which are the shorter amino acid chains, with potential bioactive properties. For example, peptides derived from caseins, such as casomorphins, have been studied for their potential positive effects on health and cancer. And we'll talk more about that shortly. However, it also appears that most of the problems we see with milk when it comes to cancer appear to be due to casein, according to the studies I've reviewed. So let's start with this one. So the paper is entitled B-casein-derived peptides produced by bacteria stimulate cancer cell invasion and motility. So let's quote from the study. A new pro-invasive peptide was isolated from an in vitro collagen type 1 co-culture system with listeria and colon cancer cells. So just to be clear, you think, well, what does it matter if I don't have listeria? Then it's not an issue. But listeria and also E. coli and also salmonella are bacteria that can be found in the human gut, right? And the presence of these bacteria in the gut does not necessarily cause sickness. So you could be walking around so you might think, well, you would know if you had listeria or E. coli. Not really, no, because God knows how many people are out there with small amounts of E. coli and listeria. You don't have to get sick from it. In fact, many strains of E. coli are part of the normal gut flora and perform important functions in the digestive system. Salmonella Salmonella typhimurium is a type of salmonella bacteria that can cause food poisoning, but it is not typically present in healthy individuals in large numbers. Listeria monocytogenes is a pathogenic bacterium that can cause a serious infection called listeriosis. And while Listeria monocytogenes are not a common inhabitant of the healthy human gut and usually are associated with contaminated food, individuals can become carriers of the bacteria, meaning they harbor the bacteria in their gastrointestinal tract without showing symptoms of illness. So the presence and levels of all these bacteria in the gut can vary depending on various factors, including your diet, including your hygiene levels, including overall health. The key is that gut bacteria have mechanisms, as shown in the study, to break down proteins like B-casein and generate peptides as byproducts. In this case, in the case of the study, the 13-mer peptide derived from B-casein was identified and found to have pro-invasive properties and stimulated the invasive behavior of colon cancer cells. In fact, according to the study, quote, pro-invasive activity was also generated with CM col of enteropathogenic E. coli and of Salmonella typhimurium, or of bacteria isolated from tumor biopsies of colon cancer patients and grown in TSB, which is CM col bacteria, tumor TSB, demonstrating that stimulation of cancer cell invasion is not just restricted to listeria, right? So it's not just listeria. They're also showing here salmonella and E. coli can cause the same thing. So now so let's look, by the way, at the rat study that they mentioned in this paper. They also mentioned the rat study, right? They mentioned this rat study. What did I say about that? Right? They said that B casein, right? B casein. Putative participation of B casein peptides in cancer development is suggested by the finding that oral administration of heat-treated casein to rats promoted the formation of colon tumors. So I mentioned that earlier. Let's look at actually the study itself, what it says. It says the study results were published in a paper entitled Promotion of Aberrant Crypt Foci and Cancer in Rat Colon by Thermalized Protein. All right. So now we're looking at another study, which again points to casein as being the culprit being a culprit in colon cancer. So, quote, we have previously shown that thermalized protein, right? So thermalized casein, which is cooked, right? Cooked casein protein with fat in the diet of the rat promotes the growth of aberrant crypt foci, which are considered to be precursors of colon cancer, assessed at 100 days after. To determine whether the previous finding of promotion of aberrant crypt foci corresponds to promotion of cancers assessed much later, we carried out promotion studies until colon cancers appeared. And the conclusion of the study was that thermalized casein promotes early colonic precursor lesions in a dose-dependent and thermolysis time-dependent manner. Essentially, thermalized casein also promotes colon cancer. Okay, so there you go. So two studies right there showing the effects of casein on cancer. Now let's go to a study entitled Milk Stimulates Growth of Prostate Cancer Cells in Culture. So, quoting from the study, concern has been expressed about the fact that cow's milk contains estrogens and could stimulate the growth of hormone-sensitive tumors. In this study, organic cow's milk and 2 commercial substitutes were digested in vitro and tested for their effects on the growth of cultures of prostate and breast cancer cells. Cow's milk stimulated the growth of LNCAP prostate cancer cells in each of 14 separate experiments. That's kind of scary. Producing an average increase in growth rate of over 30%. All right. And here's another study that demonstrated casein's pro-cancer effect entitled A Milk Protein, Casein as a Proliferation Promoting Factor in Prostate Cancer Cells, found that, quote, PC3 cells treated with 1 milligram per milliliter of A casein, so now not just B casein, A casein, and casein itself showed increased proliferation, 228% and 166% respectively. And the proliferation of LNCAP cells, right, there's a certain type of prostate cancer cell, was also enhanced by 134% and 142% respectively. The proliferation mechanism of A casein in PC3 and LNCAP cells did not appear to be related to the induction of insulin-like growth factor, since the level of IGF-1 did not change upon the supplementation of casein. That's long been suspected that, one of the reasons why the cancer could be attributed to milk is because milk increases the insulin-like growth factor hormone, which can cause tissues to grow and cancers to grow. But that's not what's being shown here. Now, to be fair, they also tested 5 other very specific cell lines, including one for breast cancer, one for lung cancer, for gastric cancer, and found that, quote, A casein and total casein did not affect the proliferation of those cells. But they did conclude that, quote, the milk protein casein promotes the proliferation of prostate cancer cells, such as PC3 cells and LNCAP cells. So again, if you have prostate cancer, should you be, concerned? about milk consumption. Something for you to think about. With all of that said, right, I don't want this to be a one-way street where I just harp on or focus on the studies that point to the damaging effects of milk and casein protein when it comes to cancer. All right, I have to be completely transparent here because I did come across a number of studies which show that that milk, actually cow's milk, has been shown to have beneficial effects. Right, so let's be completely honest here. I want to point out that milk proteins have also been shown to have anti-cancer effect. For instance, here is a paper entitled "The Milk Protein A Casein Functions as a Tumor Suppressor via Activation of STAT1 Signaling, Effectively Preventing Breast Cancer Tumor Growth and Metastasis." So, quote, right, this study that they're publishing about, quote, identified the milk protein A casein as a novel suppressor of tumor growth and metastasis. Briefly, MET1 mammary tumor cells, MET1 mammary tumor cells expressing A casein showed a roughly 5-fold reduction in tumor growth and a near 10-fold decrease in experimental metastasis. Okay, that's an amazing thing. That's a great thing. Here's another paper entitled "Growth Inhibitory Effects of Casein Hydrolysates on Human Cancer Cells," which found that casein hydrolysates B, C, and F had an inhibitory effect on the viability and growth of both human JurCat T cells. By the way, JurCat T cells are used to study T cell leukemia, and CACO2 cells, CACO2 cells, which are colon cancer cells. And so there you go. Another study show, in this case, growth inhibitory effects of casein hydrolysates on both those two different cancer cell lines. So again, excellent. And here's a paper from 2019 published in the Journal of Oncology that sums up what appears to be, from my extensive research anyway, most of the experimental or laboratory-based studies. So like there's this one paper published in 2019, which kind of combines and takes a look at a lot of the studies that have been done on casein and milk and cancer. And so this is a really good source. If you just want to read one study, or if you want to read just one paper, read this paper, published in 2019, Journal of Oncology. Right. And so most of it basically it looks at most of the experimental or laboratory based studies that demonstrate the anti-cancer properties of bovine milk derived casein. And when I say experimental or laboratory based studies, what I mean are the interventional studies that investigate cause and effect relationships, as opposed to epidemiological population-based studies, which I've left out of this podcast. I'm really not interested in epidemiological population-based studies, which are essentially they're observational. All right, so those studies are merely observational. They don't prove cause and effect, and merely they basically speculate on the possibility of causal relationships because, right. They're looking at population based studies where you're looking at like milk consumption over a period of time amongst a large group of people, and they're basically doing surveys on what these people are taking in, and then they're looking at their health outcomes over a long time, and then they're speculating on the causal relationships, but that's not evidence. All right. And look, there are also a few human milk studies in here as well. With that said, there are also a few human milk studies and if you want to get into human milk, because there's a ton of great stuff in human milk and what human milk, does to the body and the effects that it has on cancer and the body. Positive effects, right? But we're not, I don't want to get into that, because the point of this podcast is to look at why you need to be really concerned and why you need to, pay attention if you have cancer to cow's milk, okay, to bovine dairy products. So we're not, so this study is great, but it also talks about some human milk studies, right? But nevertheless, right. With that said, let's talk about if you want to read it. It's called the paper is called Casein and Peptides Derived from Casein as Anti-Leukemic Agents. And the paper starts out by stating, quote, studies in vitro suggest that caseins and the peptides resulting from the enzymatic hydrolysis of casein, that's the enzymatic digestion of casein, have anti-tumor activity, which agrees with studies in humans that show that a lower frequency of breastfed infants develop leukemia, and a similar effect has been described for older adults who consume milk of bovine origin. So again, being completely transparent, this overall paper, which points these things out, again, you got the good, but you also have the bad and the ugly. But it's also pointing out that it has been shown, not just breast milk, but it's been shown that in older adults who consume milk of bovine origin, they've seen the effect that, you've seen the effect of a lower frequency of the development of leukemia. And the paper goes on to point out examples of the anti-cancer effects of caseins and casein peptides on different cancer cell lines or on animal models, including, going to again quote these studies so you don't think I'm being one-sided here. Quote, among the first findings of anti-tumor activity of casein in vivo, that's in vitro is in the lab, in vivo is in the living organism. Among the first findings of anti-tumor activity of casein in vivo, rats fed a diet rich in casein showed a marked decrease in colon carcinogenesis that had been induced by azoxymethane compared with the carcinogenesis in rats fed a lower casein diet. Okay, interesting. The paper also references studies that show, quote, A, B, and K casein proteins all inhibit the migration in vitro, that's in the petri dish in the lab, of murine mammary tumor cells of the MET1 cell line, MET1 cell line, the human breast cancer cell line, also the human breast cancer cell line MCF10A HRAS, G12V, and the MDA MB231 cells, with A casein being the most effective. Look at that. And that peptides derived from AS1 casein and B casein digested by lactic acid bacteria inhibit the enzymatic activities of purified recombinant matrix metalloprotease MMP2, MMP7, and MMP9 in human HT29 and SW480 colon carcinoma cells. Okay, so again, there you go. And earlier, as we mentioned, right, I mentioned casmorphins and their potentially positive effects on cancer. Well, it turns out that in addition, right, if you're looking at 90 to 95 and 90 to 96A casmorphin, BCM7, BCM5, and the morphiseptin, the amide of B-casomorphin 4, all have an anti-proliferative action on T47D cells, blocking cells in the G0, G1 phase. So again, right, when you look at casein, and in this case casmorphins, you're seeing a beneficial effect. And 90 to 95 and 90 to 96A casmorphin, BCM5, and AS1 casmorphin amide all inhibit the proliferation of human prostate, DU145, and PC3 cancer cells. By the way, 90 to 95 and 90 to 96, acasmorphin, BCM7, BCM5, and morphiseptin. These are all, they're all casmorphins, okay? And casmorphins are opioid peptides that are derived from the digestion of casein, the most abundant protein found in milk, as we've pointed out earlier. And when casein is broken down during digestion, it releases smaller protein fragments. And some of these fragments are casmorphins. Casmorphins are morphiseptin, right? Sorry, casmorphins and morphiseptin have been of interest to researchers due to their opioid-like effects, which can influence physiological processes, including pain, perception, and behavior. However, it's important to note that the opioid activity of these peptides is relatively weak compared to other naturally occurring opioids like morphine. Additionally, their concentrations in milk and the human body are generally low, so they are not considered to be major contributors to opioid activity in humans from milk consumption. I do wonder, though, if you eat cheese, it's hard to break the habit of eating cheese. I wonder if there's an addictive effect there based on the stimulation of those casmorphins breaking down breaking down the casein into these casmorphins, these opioid peptides hitting the opioid receptors. I'm wondering if it's stimulating that addiction. Pretty interesting. I haven't looked into that. The paper also mentions a study that shows that PGP-IPN, so let's just call it PGPIPin, a hexapeptide derived from bovine B casein, inhibited the proliferation of human ovarian cancer cell line, SKOV3, as well as the primary ovarian cancer cells in vitro, and that consistently, PGPIPin also decreased tumor growth rate in xenograft ovarian cancer model mice in a dose-dependent manner. Further study demonstrated that the anti-tumor effect of PGPIPin is partially through promoting cell apoptosis, that's cell program death, by inhibiting BCL2 pathway. Okay. So yeah, sounds like there's a ton of evidence to support the claim that milk proteins have anti-cancer properties as well as cancer-promoting properties. However, my concern is whether someone who already has cancer should make milk and dairy a staple of their diet. Given that I made cheese a significant part of my meal plan for at least a year and a half, or at least a year, maybe. At least a year, could have been as long as a year and a half, before noticing that the tumor in the center of my kidney had spread to another part of my kidney, and, went on for another 6 months of eating cheese and adding a bit of heavy cream to my coffee for a period of time, as that second tumor increased in size. Remember, I said they found it was 1.1 centimeter when they found it. I don't know why, but I didn't think to look at cheese being the issue. It was really the only change I had made to my diet at the time. Again, ketogenic diet. Cheese, is definitely on the menu if you're on a ketogenic diet. And I didn't do my research like an idiot. I went on eating it. So not only did the tumor spread to another part of the kidney, but it kept increasing in size before I made the correlation, before, my brain switched on and said, oh, wait a second. Do you think it's the cheese that's maybe doing this? And then I did my research, found a couple of studies that I've mentioned, by the way, of the ones I've mentioned, on the negative effects of casein as related to the metastasis of cancer. And I came to the conclusion that I should cut all dairy out of my diet forever. Even though there's these other studies showing positive effects, listen, why take the fucking chance? So with that said, here is the most crucial and concerning study of them all. So even if you look at casein, you see, okay, there are some studies showing casein is bad for cancer. And then you show that there are some studies showing that that casein is good, right? So in some studies, casein is promoting tumor growth. In some studies, it's decreasing tumor growth. Let's say you've got studies on both sides. Maybe it's a wash, right? Maybe it's a roll of the fucking dice. Based on that alone, I still wouldn't eat cheese if I had cancer. I still wouldn't eat cow's milk products if I had cancer. But put the casein aside for a second. Put the milk proteins aside for a second. Put those studies aside for a second. Right? For me, the real scary one, the real scary study that's out there is talked about in a paper that's entitled Oral Administration of Bovine Milk-Derived Extracellular Vesicles Induces Senescence in the Primary Tumor, right? Senescence in the primary, which is a good thing, but accelerates cancer metastasis. Okay, so it tells you right there in the title of this paper, right, that there's a major issue. Now, before I get into the guts of this study and tell you what's at play, let's explain what the hell extracellular vesicles are. So extracellular vesicles, EVs for short, are a diverse group of small membrane-bound vesicles that are released by cells into the extracellular environment. They play essential roles in cell-to-cell communication and are involved in various physiological and pathological processes. EVs are present in various bodily fluids such as blood, saliva, urine, and milk, and are found in virtually all living organisms, including plants, animals, and microorganisms. And there are three main types of EVs, distinguished primarily based on their biogenesis and size. So you have exosomes, microvesicles, and apoptotic bodies. So apoptotic bodies are the largest of the EVs. They're usually larger than 1,000 nanometers and are released during apoptosis. So again, apoptosis, that's what happens when cells, it's a programmed form of cell death, right? So a cell recognizes that it's broken or dysregulated, mutated, problematic, whatever the case, and it based on it recognizing its condition, it goes into a cell suicide program, gets the hell out of the way, lest it cause more problems like cancer and other diseases. When cells undergo apoptosis, they break down into smaller fragments called apoptotic bodies, right? Which again, I've told you that cancer can actually use those bodies to feed, which can, right? Because when the apoptotic bodies, which contain cellular contents, including the, inside cells are little organelles like the mitochondria, the powerhouses of the cells, or little tiny organs within the cells. Right? You have ribosomes, which are the protein factories of the cells, right? So they break down into these little, pieces of the cell, DNA fragments, etc. But it's important to note that apoptotic bodies, before I go off on another tangent, are not commonly found in milk. Apoptotic bodies are generated during the process of programmed cell death, right? So anyway, these apoptotic bodies are then phagocytosed by neighboring cells, or as I've said, right? They get scavenged up by other cells, right? And in the context of milk, the presence of apoptotic bodies is minimal. Since lactation is a dynamic process of milk production and secretion rather than a process of cell death and elimination. So, sorry, I'm going off on a tangent. So milk contains mainly exosomes and microvesicles among the three types of extracellular vesicles. Both exosomes and microvesicles are released by mammary epithelial cells into milk during lactation. Now, exosomes are small vesicles with a diameter ranging from about 30 to 150 nanometers that are formed through the endosomal pathway in cells. And in the context of milk, mammary epithelial cells secrete exosomes into the milk, where they play a role in extracellular communication and the transfer of bioactive molecules from the mother to the offspring. Then there's microvesicles, also known as ectosomes. Those are larger than the exosomes, typically ranging from 100 to 1,000 nanometers in diameter, that are formed by direct outward budding and fission of the cell's plasma membrane. Mammary epithelial cells release microvesicles into the milk, and like exosomes, they are also carrying a cargo of bioactive molecules that can influence the development and the health of the offspring. So back to the study. Okay, now that I've explained ad nauseum what the hell extracellular vesicles or EVs are, let's get back to the paper itself. Let me take a sip of water. By the way, I'm sorry if I'm boring the shit out of you with all of these studies and all of this jargon. I know it's probably hard to sit through this. I probably wouldn't even sit through all this shit unless I had cancer, which I do, and wanted to learn something, which I do, right? So I've looked at these studies and I'm interested in them, even though they're boring as fuck. Why? Because I'm trying to educate myself, because I have cancer and I'm trying to keep myself healthy as long as possible. But I'm sorry if I'm boring you, and just skip over some of this shit if you want and get to the maybe the heart of the matter. So you could always fast forward me. But back to this crazy study, right, which is telling you extracellular vesicles can cause cancer to metastasize, which you definitely do not want. Quote, among mammalian dietary sources, milk is one of the most highly consumed beverages, containing an abundance of EVs. Milk is a uniquely complex epigenetic imprinting system. That's what you're drinking, people. An epigenetic, what is epigenetic? If you've listened to my past podcasts, you'll know that epigenetics means that there are certain factors that can influence the way genes turn on and off, the way genes turn on and off to create proteins. Milk is uniquely, so sometimes genes get mutated and become cancerous, but sometimes environmental factors can cause genes to switch on and off and create proteins in a dysregulated fashion that can cause cancers. That's what epigenetics means. Okay, well, it doesn't just mean that for cancer. It just means that, could switch. Epigenetics is the process of the environment influencing certain genes to switch on and switch off and create certain proteins that do certain things in the body. Okay. But that's what you're drinking, people. A complex epigenetic imprinting system that has evolved to specifically support postnatal growth and has had remarkable impact on human growth and development. Being an immensely intricate secretory product, a product that gets secreted from an organism, specifically designed for regulation of neonatal development, its persistent consumption throughout the lifespan, not just during infant development, but throughout the lifespan, may have substantial implications on human health. What does that mean? I don't know. Maybe it means that milk is safe to be consumed when you're developing as a baby. But should you be drinking it long into adulthood? Maybe not. Right? Maybe not. Back to the study, back to the paper. Both raw as well as commercial milk have been shown to contain extracellular vesicles that carry a cargo rich in RNAs and proteins. EVs confer protection to the luminal cargo, the proteins and the RNA, against harsh degrading conditions. And it has recently been demonstrated that milk-derived EVs have been taken up by human and murine gut epithelial cells in vitro. So first off, this study showed that milk-derived EVs can survive the harsh conditions. of the gastrointestinal tract after oral administration and be bioavailable in various organs, demonstrating the potential for cross-species communication. Now, cross-species communication, in this instance, people, refers to the ability of milk-derived extracellular vesicles from one species, right, cows, to interact with and influence biological processes in another species, mice. And the study demonstrated that when mice were orally administered milk-derived EVs from cows, these vesicles were able to survive the harsh conditions of the GI tract, the gastrointestinal tract, and be absorbed into the circulation. Once in the bloodstream, the milk-derived extracellular vesicles could travel to various organs in the mice and affect cellular processes. It's fascinating stuff. This phenomena of cross-species communication, by the way, along with their ability to survive the gastrointestinal tract's harsh environment due to their lipid bilayer membrane, which protects their cargo of RNA molecules and proteins from degradation, along with their ability to transport and deliver biologically active molecules directly into the cytoplasm of distant cells, including cancer cells, makes EVs very attractive candidates for potential therapeutic applications, such as drug delivery systems. Also, right, milk-derived EVs are considered natural carriers due to their biocompatibility and low immunogenicity. And they can be isolated from milk, by the way, without causing significant immune reaction or toxic effects, which is a good thing. EVs are taken up by recipient cells efficiently, facilitating the delivery of their cargo directly into the cytoplasm of the target cells. So EVs have the potential to cross physiologic barriers, such as the blood-brain barrier, which is a significant challenge for many drugs in treating diseases affecting the brain. So listen, people, the stability of EVs, the protection they provide their cargo en route to delivering their payload, the bioavailability and uptake of EVs. I mean, for these reasons, EVs, you can imagine, are being exploited as drug delivery vehicles. And that quote, sorry, I went off, going off on a tangent. What I was just saying for a fair bit of time there wasn't from the study. It was just my observations from the study. But now I'm going to go back to the study and quote, recently, milk-derived EVs packaged with chemotherapeutic drugs have been shown to reduce the primary tumor burden. So they're showing that these EVs can be a very, very powerful tool against cancer if you pack those little suckers with some poison. And then instead of doing what they do today, where you take in the poison and it just decimates the entire body, if you package it in a little bioavailable transport system, like extracellular vesicle, which has a lipid bilayer which protects it from the harsh environment of the gut and can deliver it to tissues and to cells, particularly cancer cells. You protect the rest of the body from the poison, but then when it gets uptaken by the cancer cell and delivers its cargo, boom, explosion, death, good night, Irene, to the cancer cell. Hey, that's a good thing. And, but if we get back into the paper, right, it's telling you here that, there are some good things with EVs. And, this is something that's being looked at, using EVs for their cargo carrying potential. In fact, in a paper entitled Bovine Milk-Derived Exosomes for Drug Delivery, which was published all the way back in 2016, the authors describe a study that demonstrated how rare mature bovine milk can serve as a biocompatible and cost-effective source for harvesting bulk quantities of exosomes, and that milk exosomes have tremendous potential as a drug carrier for hydrophilic and lipophilic agents, including chemodrugs. This nano device tech, it can overcome the limitations associated with the poor oral bioavailability of chemopreventatives and chemotherapeutics and lower the total administered dose, thus minimizing or eliminating toxicity generally associated with high doses. Exosomal formulation of drugs can not only enhance biologic efficacy and exosomes functionalized with ligands for tumor targeting can further improve specificity and estimate and eliminate off-target side effects of drugs. However, again, from the study here, however, further studies are warranted to rule out any potential toxicity with long-term use of milk exosomes. Okay, so very powerful information, coming from this study, bovine milk-derived exosomes for drug delivery, right? Very powerful nano device technology that nature is providing us with. Okay, so it's got some good things attached to it, right? So let's now look at, all right, here, I just want to blow this up, blow this information up here. Okay, so, all right, good, because I've got some more study information here to impart to you all. Because beyond its potential as a way of enhancing the targeted uptake and delivery of cancer-fighting drugs, extracellular vesicles appear to have intrinsic anti-cancer properties. For example, in a paper entitled "The Therapeutic Potential of Milk Extracellular Vesicles on Colorectal Cancer," quote, cow milk-derived exosomes were reported to have an inherent anti-cancer potential by reducing the growth of malignancies, such as colon and ovarian tumors. The study that they cite, by the way, in this paper is entitled "Exosomal Formulation of Anthocyanidins or Cyanidins Against Multiple Cancer Types." So the paper goes on to say that, quote, "We first determined that the anti-proliferative effects of milk exosomes per se, in the absence of any drug, on cancer cell lines of lung, prostate, colon, breast, pancreatic, and ovarian by MTT assay, the treatment of these cell lines with exosomal proteins resulted in an 8 to 47% cell growth inhibition. Survival of lung cancer cells was reduced. The lung cancer cells, I'm going to try to spare you the jargon. I'm aware that I'm boring the fuck out of you. So I'm going to try to cut out some of the jargon, and I don't need to name the cancer cell lines that they used here. Let's just say that survival of lung cancer cells was reduced by 66% and 76%, respectively, depending on the cell lines. Breast cancer cells showed cell survival. So breast cancer cells showed cell survival of 55% and 76%, respectively. Both pancreatic cancer cells that they used in the study showed nearly 65% cell survival. Prostate cancer cell lines were the most sensitive with 53%. And what was it? Also, yeah, again, another prostate cancer cell line were least affected with 92% survival. So you have all these different survival rates that they showed, right? The only ones that showed the best survival when being treated with, these, and this is without any drugs, right? Just the milk exosomes, showed that survival was reduced. And in all these different cases, some cases 66%, 76%, depending on those were the lung cancer cells, all the way down to the colon cancer cells, survival was reduced to nearly 68%, right? And the ones that were least affected, it showed that the most sensitive were prostate cancer cells, which were survival was reduced by 53%. And there was a certain prostate cancer cell line, PC3 cells, were least affected with 92% survival. Okay. Nevertheless, back to the paper, the therapeutic potential. So again, I want to be fully transparent. Where these, in this case, milk exosomes, even without drug delivery, have shown a positive effect on reducing cancer cell survival. But there's this paper, The Therapeutic Potential of Milk Extracellular Vesicles on Colorectal Cancer. They go on to talk about the anti-cancer properties of camel milk and camel milk extracellular vesicles. And this I found fascinating, and it's something I'm going to look deeper into, because put aside cow's milk and the potential concerns if you have cancer that you might have with cow's milk. There's a study out there, and actually there's other studies, so I'm not going to talk about all of them here, but there's this study on the therapeutic potential of the EVs from camel's milk on colon cancer. Quote, camel milk and its contents have been shown to have anti-tumor impacts on the liver, right? On liver cancer cell lines, on breast cancer cell lines, and mouse hepatoma. Those are liver cancer cell lines. And one of the studies they cite in this paper, which funnily enough talks about the therapeutic potential in colorectal cancer cell lines, but it says that camel milk modulates the expression of aryl hydrocarbon receptor regulated genes, CYP1A1, NQO1, and GSTA1 in murine hepatoma, murine, that's, that's mouse cancer cell lines, IC1C1, C7 cells, which showed, quote, evidence of the ability of camel milk to significantly inhibit the induction of CYP1A1, a cancer-activating gene, is strongly correlated with increased incidence of several human colorectal and lung cancers, and to induce NQO1 and GSTA1 cancer-protecting genes in HEPA1C1, C7 cells. Very, very interesting. So CYP1A1, which is strongly correlated with increased incidence of several human colon, rectal, and lung cancers, and it was shown that camel's milk in this study significantly inhibited the induction of that cancer activating gene, which again is strongly correlated with the increased incidence of several different types of cancer. And it also seemed to induce NQO1 and GSTA1 cancer protecting genes in the liver 1C1, C7 cells in the mouse. Okay, sounds pretty powerful. Camel milk suppresses cell development and causes apoptosis in HepG2 and MCF7 cells. So again, from the study, right? This is now, right? Am I going back now? I'm going back to the paper, The Therapeutic Potential of Milk Extracellular Vesicles on Colorectal Cancer. Back in that paper, okay, they show that camel milk suppresses cell development and causes apoptosis in HepG2 and MCF7 cells by stimulating caspase-3, in addition to the death receptor DR4, and accumulating superoxide radicals inside the cells. So again, just another finding that they're pointing out in this paper, where they get into the power of extracellular vesicles on colorectal cancer. Again, this is one of those papers that just shows overall, it has a ton of studies that they're citing in here about the therapeutic potential of milk extracellular vesicles. And even though the purpose of the paper is to show its beneficial effect on colorectal cancer cells, it's got all these other studies showing that it's beneficial in other types of cancers, right? So Furthermore, let's get back here. Furthermore, after studying camel milk and exosomes derived from camel milk, it was also found, again, we're in this paper, it was found that they suppress MCF7 breast cancer cell proliferation, which is accompanied by a decline in MCF7 cell migration. Boom. Rats that had cancers showed significant improvement after the use of milk, of camel milk, and exosomes derived from camel milk. Camel milk, as well as its derived exosomes, fundamentally diminished cancer weight, stop tumor development, and improve the immune system. This is I'm going to drop this, a link to this paper in the podcast show notes. It's a very powerful paper to read. Even though I understand, look, this podcast is about the damage milk can potentially do you if you already have cancer. That's pointing out cow's milk, which is what everyone drinks. But what I found in my studies, showing the good, the bad, and the ugly, on the good side, looks like camel's milk is very powerful. So back to this paper about its therapeutic potential in colon cancer. And again, all the studies it's citing on its way to showing that therapeutic effect on colon cancer cells. It's showing all the other studies that have been done on camel's milk and other types of cancers. We have here, it says that while exosomes have a greater anti-cancer impact in general, the number of splenic T lymphocytes in rats given camel milk increased significantly, demonstrating that there are more immune stimulating elements in camel milk compared with the exosomes. So compare with the exosomes alone. And exosomes derived from camel milk had a specific anti-proliferative impact on tumor HEPA-RG cells, again, HEPA, liver cancer cells, but no toxic impact on regular liver cells, regular liver THLE2 cells. The anti-tumor impact may well be related to the stimulation of apoptosis, as well as the prevention of inflammation and angiogenesis. Wow, 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 wow. So listen, all of that sounds amazing, people. And I am definitely going to look deeper, much deeper into, I might even do a podcast on the anti-cancer effects of camel's milk. However, if we return to bovine milk, especially given that 81% of the damn world drinks not camel's milk, but cow's milk. Well, then, according to the OECD, right, there's this report, the OECD FAO Agricultural Outlook 2020-2029 report, which tells you that 81% of the world is drinking cow's milk. And because of that, right, I want to focus back in on what cow's milk can do to you if you have cancer. And even though there's been good studies about the casein and bad studies on what casein can do to cancer, right, there is this, for me, this ultimately, really, really powerful study. If you're looking for something that should give you a warning to maybe consider staying away from cow's milk if you have cancer, then it's the one I've been mentioning here earlier, which is, again, just to remind everyone, if you've been skipping over a lot of the jargon I've been saying, oral administration of bovine milk-derived extracellular vesicles induces senescence in the primary tumor, again, good thing, but accelerates cancer metastasis, right? Then here's the thing. If we go back to that paper, as good as the other stuff sounded about the casein, where we're showing good things about the casein, as well as bad things, unfortunately, as promising as the milk-derived EVs are, when it comes to maybe using them as targeted drug delivery systems, we might want to consider that the study discussed in the paper showed that milk-derived EVs had implications for cancer progression. In fact, the researchers found that oral administration of milk-derived extracellular vesicles in mice with pancreatic cancer led to an increase in liver metastasis, meaning that the cancer moved from their pancreas to the liver. And similarly, in a breast cancer model, milk-derived extracellular vesicles accelerated lung metastasis, meaning it moved from the breast to the lung. And this result, say the researchers, was completely unexpected, as it was initially hypothesized that milk-derived EVs might attenuate, that means stem, the spread of cancer. What these researchers also observed was that when cancer cells were treated with milk-derived extracellular vesicles, they entered a state of cellular senescence. So this means that the cancer cells stopped dividing and their growth was arrested, leading to a reduction in the size of the primary tumor. A wonderful thing, right? So that observation suggests that milk-derived EVs have an anti-tumor effect on cancer cells by inducing senescence, which is generally considered a positive outcome in cancer therapy. However, the study also found an unexpected and counterintuitive result. Despite the induction of cellular senescence and the reduction in the primary tumor size, milk-derived EVs still led to an increase in tumor metastasis. Additionally, the study revealed that milk-derived EVs induce the process of epithelial to mesenchymal transition, known as EMT, in cancer cells. And EMT, by the way, is a process in which epithelial cells acquire properties that allow them to become more invasive and migrate more effectively, promoting the spread of cancer. And although the researchers in this study speculate that milk consumption may be beneficial to breast cancer patients when the primary tumor is once the primary tumor is surgically removed, the extremely crucial point to make here is that although the oral consumption of milk-derived extracellular vesicles decreased the primary tumor growth, it, quote, paradoxically increased lung and liver metastasis. And that hot, direct quote here, people, high consumption of milk may increase the metastatic burden in cancer patients with intact primary tumors. Okay? And so why is it doing that? Why is it doing that? It's lowering the primary tumor, but it's causing the damn tumor and the damn cancer to spread. So one of the outstanding questions in the study relates to the identification of the precise protein RNA cargo that's in the extracellular vesicle, that's instrumental in the opposing effect of reduced tumor burden with accelerated metastasis. And you can imagine that's a very important question, because if they're going to want to use the extracellular vesicles to deliver drugs, they're going to want to figure out, okay, well, which protein, which RNA, which piece of the cargo that's coming with the extracellular vesicle is actually causing the cancer to spread, because ideally they'd want to identify it, isolate it, extract it, and just use, right, that nanoparticle, that extracellular vesicle, for its ability to survive the gut, and it's for its ability to not freak out the immune system, because it's kind of a natural nanotechnology. It's kind of a natural nanotransport system to stick some drugs in, some poison in, and have it travel to the cancer cell, kill the cancer, right? But I hope that I've gotten my point across to everybody, and that you've managed to last all the way to the end of this podcast, because even for me, reading some of this jargon is a bit boring. But again, I don't care if it's boring. The point needs to be made, right? I don't want to just sound as if I'm talking off the top of my head. I want to quote the actual studies. I want to bring in the salient details. I want you to know that there's science here when I talk about the fact that milk, cow's milk particularly, can have real detrimental effects. Yeah, it can have good effects as well, has been shown in some of the studies, but it's showing that casein can have really fucked up effects. But then all of those, even if you wash all of them, I want you to understand from the studies that, for me, the big smoking gun is these extracellular vesicles. And as good as they can turn out to be, maybe for drug delivery and also for cancer, the ultimate takeaway here for all of you, I hope, is the quote from this one study on extracellular vesicles coming from cow's milk. And I'll say it again. High consumption of milk, right? High consumption of milk, and they're talking about milk from cows here, may increase the metastatic burden in cancer patients with intact primary tumors. What does that mean? To me, it's quite simple. If you have cancer, right? If you have a tumor, stay the fuck away from milk. Be warned. Be warned, right? In my opinion, stay the hell away. Look, I can only speak for myself. I'm not drinking milk. I'm not drinking cow's milk. I'm not having cheese. I'm not having yogurt. I'm not having no matter how good it is for you. I'm not having it. All I need to see is that one quote. And that's enough for me to say, hey, I don't need milk. It's all good. All good. We're not talking about an epidemiological study, right? An epidemiological study, a population-based study like that bullshit China study, which says that meat causes cancer, right? Which is a population-based study. Correlation does not equal causation. No laboratory study, right? Show me a laboratory study, right, that shows that. You're right. Show me a study where they take a human, feed the human meat for a period of time, right, locked away in a lab, right, and the meat all of a sudden causes you to develop cancer, right? What I'm saying here is that high consumption of milk, quote, may increase the metastatic burden, meaning if you have cancer, stay the fuck away. I've been eating meat for 7 years now. And I've been on a mostly meat diet for many, many years. Okay. The 2 times the cancer spread is when I ate peanut butter and when I ate milk products, cow's milk products. Now, right, correlation, which I'm making, does not equal causation, and I fairly admit that. Which is why I'm not necessarily going to take a population-based study into account. I'm going to look for evidence, right, before I make a decision. Nevertheless, when I look at this study, and the study shows you, right, that these EVs cause cancer to metastasize, and you get a quote like this: high consumption of milk may increase the metastatic burden in cancer patients with intact primary tumors. Why fuck around? Right? Why mess around? So if you know someone who has cancer, send them the podcast I did on peanut butter. Get that information to them. Get the information to them. Let them hear that so they can make their own decision. Have them look at the podcast show notes so they can look at the studies and look at the links. If you know someone who has cancer, send them this podcast, right? Tell them to be cautious when it comes to eating cow's milk and cow's milk products, right? Show them the show notes. If you don't want to listen to me bullshit for an hour and a half and listen to all the boring shit I'm spewing here, then just have them look at the show notes and look at the studies, or fast forward through the podcast to get to the interesting stuff. Nevertheless, I'm signing off now. I've talked enough. I hope, again, I didn't bore the hell out of you. But victory can be achieved with insight. This is the Alpha Human Podcast. This is Lawrence Rosenberg. Right? We continue to fight. We continue to pray to Jesus Christ. We continue to look for insight and discernment from God in heaven. God bless all of you. May Jesus Christ bless all of you. May you find Jesus Christ. May you find solace and hope and direction and guidance and solutions through Jesus Christ, and in Jesus Christ's name, through Christ, hopefully I can provide some direction and guidance and insight to you through the information, not the opinion, right? Forget about the opinion. Through the studies that I am quoting, hopefully I can impart some knowledge to you that you can use either for yourself or for a friend, so that they can be helped. And again, ensure that your friends are listening to their doctors. Ensure that your friends, loved ones, and family members, if God forbid they should have cancer, are seeking out second and third opinions. Hopefully they are also being encouraged to read studies and learn about their cancer, because you will achieve understanding, you will achieve knowledge, and you will achieve insight that could help you in ways you may not even understand now, that are life-affirming and that are life-sustaining through insight. Victory through insight, people. This is Lawrence Rosenberg signing off. Have a great day. Have a great week. See you next time.